Today we are going to look at different stages of embryo, and here I have the different stages of embryos kept in this liquid medium in this, but you cannot able to see the actual embryo in it. And with this lab we are going to use an in a microscope called as dissection microscope, which is little different from the other microscope. Basically your microscope, normal laboratory microscope, will have the light source and followed by the specimen and followed by the lens and the eyepiece, whereas the dissection microscope will have not the light source but will have a mirror, which I am showing it here. So you have to adjust the mirror in such a way that the external light is reflected into the mirror and it has to be passed on to the specimen stage. This is the specimen stage, where we are going to keep the embryos which are already there in the petri plate, and we are going to keep the petri plate on top of the specimen stage, and we have to adjust this mirror in such a way that the light are reflected onto the mirror and it is being reflected onto the sample. Okay, the samples are here, but naked eyes we cannot able to see that, but after looking at in the microscope we can able to see that. And another important difference in this microscope, the dissection microscope, is the we have only one adjustment called as coarse adjustment, whereas in the case of normal microscope, laboratory microscope, we have coarse as well as fine adjustment. But this microscope, dissection microscope, has coarse adjustment. By doing so, the adjustment, you can tend to see that the lens goes up and down. Okay, so this is almost equal to adjusting the image to make a clear vision. Okay, and we have only one lens, that is 10x lens, and with the help of this 10x lens you have to keep your eye on the 10x lens, and then you have to adjust it in such a way that you can able to clearly see the image or the visual of the specimen that is present in this petri plate. Okay, so now I am adjusting the image in such a way that I can show you through this lens how the embryo is going to look at.
Students, we had observed how to use this dissection microscope, and now we are going to see the actual different stages of embryo. Now we are going to visualize the different stages of developmental stages of embryo of a zebrafish. And if you look at it, this image, you can see that there are 8 different stages of embryo developments are there, but you can actually see 5 stages, starting from zygote, cleavage, blastula, gastrula, segmentation. Followed by that we have pharyngula, hatching, and then early larva stage, which we'll be seeing little later. Now coming back to the importance of different stages of embryo is that at every single stage there is a unique transition that is going to take place. And in the case of zygote, as soon as the eggs are fertilized, it is going to be converted into a zygote stage. Followed by that we have cleavage. Okay, during the cleavage we have the cell cycles that are going to happen, and if you look at the cell cycle that is going to happen during the cleavage stage is going to be little different from the one that is regularly happening in the normal cells. The cell cycle that occurs during the cleavage is going to be really short, in the sense there is only short synthesis phase followed by that mitosis phase. There is no G1 and G2 phase that occurs during the cleavage stage. So this cell cycle is going to be really rapid, and it is going to be synchronous. Synchronous means every single change happens in each of the divided cell is going to be the same. There is no difference between one cell and the other cell during the cleavage stage. Followed by that we have blastula stage, and here it is going to be rapid. Metasynchronous cell cycles is going to take place, and this is the place where the epiboly will start to begin. And epiboly is the kind of a movement that will be seen only during the development stage is going to be gastrula. It is also sometimes referred as gastrulation, wherein morphogenetic movements of involution, convergence, and extension from epiblast, hypoblast, and embryonic axis is seen through the end of epipole. Followed by that, we have segmentation, wherein the pharyngeal arch and neuronal development, primary organogenesis, and earlier movement in the tails appears at the particular stage called as segmentation. This summarizes the first five stages. Now we have the sixth stage called pharyngula, and in this stage we tend to see that the body acts as statins from its early curvature like structure, and you can still notice that there will be an yolk sac will be present, and during this stage there will be a pigmentation start to appear, fin will start to develop, and the circulations will also start to kick in. Followed by that, we have the next stage called as hatching. At this stage we tend to see that there is completion of rapid morphogenesis of primary organ systems. Cartilage development will be seen in head and pectoral fin, and now the hatching will occurs asynchronously, meaning it is not that all the eggs will hatch at the same time. It will hatch at different point of time, and that is what we call as asynchronous. Last stage is referred as larval period or early larva, wherein you can tend to see that the yolk sac still remains, but it will disappear soon. Along with that, it will be having a swim bladder which is going to inflate, and at this point of time it is going to be food seeking organism, and also acquires active avoidance behavior, which is a typical feature of a very recent hatched uh, zebrafish. So to conclude this virtual laboratory session, we had discussed about 8 different stages of developmental stages of zebrafish, and every single stage has its own peculiar features. Thank you students for watching this virtual laboratory session.